The overall goal of the following experiment is to acquire micron-scale three-dimensional images of the oxygen concentration in the immediate environment of live cells by electron spin resonance, or ESR, microscopy. The protocol will be demonstrated on cyanobacteria, which are common photosynthetic cells. This is achieved by first preparing a sample of cyanobacteria cells inside the ESR microscopy sample holder embedded in stable organic radical solution. The sample holder is placed inside the imaging probe, which goes into the static electromagnet of the ESR microscope system. Now, several three-dimensional ESR microimages of the organic radical solution are acquired by the ESR microscope. This is carried out for several variants of pulse sequences and often under various external conditions, such as under light and dark conditions. Next, the raw data images are processed by the computer in order to extract the three-dimensional spatially resolved relaxation times of the radical. These relaxation times provide a direct measure to the oxygen concentration through pre-existing calibration, leading to full three-dimensional oxygen maps of the measured samples under various external conditions. Results are obtained that clearly show the lack of oxygen production by the cyanobacteria when no light is applied. Furthermore, it is shown that under light irradiation, significant oxygen gradient and oxygen heterogeneity are created near the cyanobacteria cells.
Hi, I'm Revital from the laboratory of Aharon in the Schulich Faculty of Chemistry at the Technion, the Israel Institute of Technology in Haifa. Today we will show you a procedure for mapping oxygen concentration near live cells in a three-dimensional manner at high spatial resolution by the means of